Hello everyone, I will give a brief introduction to this review. This review witnessed major advances in the crosstalk between intestinal stem cells and gut microbes. Intestinal epithelium undergoes rapid cellular turnover relying on the local niche to support intestinal stem cell function and self-renewal. Research into association of intestinal stem cells and diseases continues to expand at a rapid rate. However, the detailed interaction of intestinal stem cells and gut microbes remains to be clarified. Thus, it is necessary to further explore the regulation of gut microbiota on intestinal stem cells. Furthermore, these major advances collectively drive us ever closer to breakthroughs for regenerative medicine and cancer treatment by microbial transplantation in the clinic. The context of this review is based on the fact of intestinal diseases. Many immune cell-related diseases such as inflammatory bowel disease was affected by a wide range of factors. In addition to humans, animals also have serious intestinal diseases. For example, livestock and poultry such as pigs, chicken, cattle, and sheep suffer from diarrhea and intestinal inflammation throughout the breeding cycle. Although the precise etiology of these diseases remains unclear and controversial, it is clear that activated and accelerated intestinal stem cell proliferation play a central role in recovery of intestinal integrity. Intestinal stem cells play a central role in governing rapid and perpetual renewal of the mammalian intestinal epithelium. The intestinal epithelium consists of villi and crabs. Intestinal stem cells that reside at the bottom of the crabs direct the consequent self-renewal of intestinal epithelium. Function of intestinal stem cells closely depends on the microenvironment they reside. The microenvironment is referred to as the intestinal stem cell niche, and in the small intestine, the niche can be roughly divided into three compartments: differentiation compartment, TA cell, and stem cell compartments. An LGR5 positive intestinal stem cell are located at the bottom of the craft with panic cells, while BMI1 positive cells stay right above the panic cell. And in the large intestine, villi are missing and crabs located in the base of the intestinal structure become the main location of intestinal stem cells. The normal operation of niche is achieved by regulation of several signaling pathways. The Wnt, BMP, and Notch signaling molecules co-modulate stem cell function. First one, Wnt, is the primary signaling pathway that drives intestinal stem cell proliferation and decreases differentiation in the niche. In this process, activity of Wnt gradually decreases along the craft-villus axis and reaches a minimum at the villus, which is the exact site for differentiation. Moderate activation of the Wnt signaling pathway induces intestinal stem cell proliferation and self-renewal. And the BMP signaling pathway negatively regulates self-renewal, replication, and proliferation of intestinal stem cells, thus preventing increase of their numbers and fission of crabs and promoting cell differentiation. Activation of the Notch signaling pathway is decisive in promoting intestinal stem cell proliferation and restricting their specification. Receptors, membrane-bound ligands, and DNA-binding proteins constitute the main component of the Notch. Unlike other stem cells, intestinal stem cells are contacting with intestinal microbes. A dense and diverse community of microbiota play a central role on several key physiological functions of the host. The intestinal microbiota, together with a diverse array of metabolites, interact closely with intestinal stem cells. intestinal stem cell differentiation affects gut microbes. And meanwhile, the presence of intestinal microbes also regulates intestinal stem cell renewal rate. The specific microbes listed in the above table are all involved in the regulation of intestinal stem cells in a direct or indirect manner. Within the intestine, neurophages and macrophages recognize and respond to changes in microbiota via pattern recognition receptors. Pattern recognition receptors, particularly toll-like receptors and knot-like receptors, are not limited to sensing pathogenic invasions or epithelial injury. They are also capable of regulating intestinal stem cells. Toll-like receptors are capable of regulating crosstalk between the host and gut microbes. Source identified microorganism-associated molecular patterns of symbiotic bacteria, and the interaction among gut microbiota, toll-like receptor signaling, and intestinal stem cells may exist via the Wnt and Notch pathways. Notch2 is a vital member of knot-like receptor subfamily, which is important for recognizing bacteria and sustaining the intestinal microenvironment. Microbial components, or metabolites, which we call bacteria-related products, collectively always serve as significant mediators in the crosstalk between intestinal stem cells and microbes. Tryptophan maintains intestinal stem cell homeostasis through aryl hydrocarbon receptor. In the gut, the three major tryptophan metabolism pathways carried out under direct or indirect control of the microbiota. Tryptophan metabolites regulate aryl hydrocarbon receptor through serving as its ligands, and short-chain fatty acids are identified as intestinal stem cell effectors. Butyrate suppresses intestinal stem cell proliferation at physiological concentrations. However, cleft structure and corner size can protect intestinal stem cells to alleviate this effect. In conclusion, intestinal homeostasis depends on the dynamic balance between intestinal stem cell self-renewal and differentiation. Factors including the niche, intestinal microbiota, metabolites, and immune systems co-regulate physiological activity of intestinal stem cells. So in this review, we focus on intestinal stem cell niche and our current understanding of the role, regulation, and intervention of microbiota in this process. The understanding of microbial regulation and intervention in this process has the potential to be the therapeutic target involved in intestinal diseases. Considering that the detailed interaction of intestinal microbiota and intestinal stem cells remains puzzling, we hope the content in this review can fill the gap and provide a useful and topical contribution to the intestinal stem cell field.